region. And I like to welcome Nikki Noren Hooten, who's been kindly agreed to co-host this talk today. So today we have a kind of special topic here. We have Christina Schuh from Chile. Christina is a biomedical engineer with a PhD in technical sciences from the University of Technology, Vienna, Austria. She started working on extracellular vesicles during her second postdoc at the Universidad de los Andes, Chile. I hope I read it correctly there. where she led a project on the transfer of EVs from laboratory into the clinics. And during this time, she got interested in the topic of extracellular vesicle interkingdom communications, on which she is currently working as an assistant professor at the Universidad del Desarrollo, Santiago de Chile. So today, the topic of our talk presented by Christina Schuh is exploring new kingdoms, honeybee-derived extracellular vesicles and their potential in wound healing. So please welcome Christina.
Thank you very much for the kind introduction, Carolina, and thank you very much for the invitation as well. I'm very happy to be here and to speak on my new topic, which is exploring new kingdoms and how honeybee-derived extracellular vesicles develop their potential in wound healing. So as you've mentioned, a second. Yeah. I'm originally actually from Austria. I'm not Chilean, and it's a long way from Vienna to Chile. So I did my master's and my PhD at the LBI trauma in Vienna and at the Technical University as well, where I was actually involved in nerve regeneration and stem cells. So then I did my postdoc at the University College in London, where I was again involved in nerve regeneration and biomaterials. And then I moved to Chile. Sorry, can't go back. Yeah. I did an industry year at the Universidad de los Andes, together with Cells for Cells, which is a startup company in Santiago. And there I was for the first time involved in exosome isolation techniques, transfer of exosomes into the clinics, and it was really fascinating. And I have to say, Chile is not just a very beautiful country to visit for vacation, but it's also a country that has a lot of amazing universities and a lot of really high-tech universities that are really interesting to work at. So I'm currently at the Universidad del Desarrollo. This is our campus, it's our new campus. We're building many buildings at the moment there. This is my center, the Centro de Medicina Regenerativa, where I'm an assistant professor at the moment. And this has been my team, my BEXO team in the year 2020. It was one of the first times we actually met in the year 2020 because of the pandemic. So going straight into the topic. We all know about classical sources of extracellular vesicles, and that is cell culture. We all know, we take cells, cell lines, primary cells, we put them in cell culture, we wait for the cells to liberate exosomes, and those exosomes we then isolate using usually ultracentrifugation. What we can also do is tissue culture, and then we're going into the more fancy sources, which are blood and plasma, saliva, urine, and recently it has also been shown that milk contains extracellular vesicles. So those are the main sources that most people would know about. But not only mammals have or produce exosomes. It has been shown that basically everything that has a cell can make an exosome. Yeah, so that includes insects, that includes bacteria, that includes fungi, and that also includes plants, which has been shown very recently. So when we think about exosomes and their function, it's usually for communication within the body of an animal or a human, within the body of a plant. Yeah, but it has also been shown in the last couple of years that exosomes are actually facilitators of interkingdom communication. That means that a plant exosome or extracellular vesicle can actually communicate with a mammal and have an effect. And also insects or reptiles, bacteria, they all can communicate in one way or the other with different kingdoms. It's actually a figure from a review we were writing a couple of years ago. And we were looking into insect extracellular vesicles. And I found that, interestingly, there's absolutely nothing on bee extracellular vesicles. And that for me was a bit surprising because I remember that was me in 1990 in Austria when I fell off a bike. Imagine I was very clumsy at the time. And that represents my mom. My mom always used to put honey on our wounds. And she used to say, so it doesn't get infected. As a kid, I was in a lot of pain. I had no clue what an infection was, but I knew that honey helped me. Growing up, of course, you start thinking about, but how these natural remedies really helping? Is there something to it? How does it hold up with the science? So I started becoming interested in honey around a couple of years ago. And started looking around. And what I found was that different cultures, independent of where they're from, where they were located, it was the Celtics, the Egypt, the Mapuche here in Chile, Campo, TCM, Ayurveda, you name it, all the cultures that had access to honeybees used honey for very similar things. And that was treating infections, treating fevers. It was used by everybody for skin conditions, for wound healing, or also as a vehicle for other drugs. Yeah? So think about it, that independent of where in the world you encounter honey, all the cultures use it for the same thing. So being a scientist, you have to also look into the literature, where it actually holds up in an in vitro setting or in an in vivo setting, preclinical setting, clinical setting. And actually there's a couple of publications that state exactly that honey is antibacterial, that honey helps with wound debridement and with reepithelialization of wounds of all kinds. And also looking into other bee products, because honey is not the only bee product that is available on the market. There's also royal jelly and bee pollen. I'm going to explain in a minute what the difference is between them, which has also been shown to be antibacterial. They have been shown to be anti-inflammatory. Royal jelly has been shown to be very interesting for improving mucositis in a clinical setting. And bee pollen has been shown to improve burn wounds. So the question is, what do all these three have in common? All these three are in one way or the other associated to the so-called hypopharyngeal gland. The hypopharyngeal gland, which has the worst name in the history of any gland, is located in the bee's head here. And it's one of the most fascinating organs I've encountered. The reason for that is it absolutely changes its function over the lifespan of a bee. So it starts off with young nurse bees. Young nurse bees do not leave the hive. Young nurse bees stay in the hive and are responsible for the nutrition of the bee queen and of the nutrition of the young larvae. So they produce royal jelly, which is completely in the absence of any kind of plant product, and it's only produced by the nurse bees. As soon as the bees get a bit older and start foraging, which is around 17 days of their lifespan, they start to collect nectar and bee pollen, and nectar, with the help of the hypopharyngeal gland and a number of other enzymes, is converting, nectar is being converted into honey by the worker bees. Then also bees are collecting pollen, which is compacted with the help of saliva and with the help of the hypopharyngeal gland. And this bee pollen, again, is activating the hypopharyngeal gland of younger nurse bees. So it's a full circle. Why did we think that bee products would contain extracellular vesicles? Many glands have been shown to produce highly active extracellular vesicles. So for example, the human salivary gland has been shown to contain extracellular vesicles, different types of extracellular vesicles, and those could contain different enzymes, like deep peptidase, ERM proteins, tissue factor, venom. Extracellular vesicles have also been shown to be very active in the absence of any other compound. Snake venom, for example, of the copperhead viper, has been shown to contain extracellular vesicles, which are highly active and can actually change actin organization. So we thought, well, all these products have some relation to this gland, so they probably contain extracellular vesicles as well. So the only way to know it is to try it. So we tried to develop an isolation protocol for bee pollen, honey, and royal jelly-derived extracellular vesicles. And what we did was the following. We had the raw product, dissolved it in particle-free PBS. This underwent serial centrifugation, filtration, and then ultracentrifugation. What we used here was a swing bucket centrifuge of a Sorval WX thermoscientific centrifuge, and we centrifuged for 70 minutes. Of course, we have to quantify right afterwards if we actually got particles in the vesicle range that we're looking for, which is around 50 to 150 nanometers. And using nanoparticle tracking analysis and the Malvern nanosat NS300 under temperature-controlled conditions, camera level 8, threshold 3, and as a control particle-free PBS. We found that all three sources, all three raw products, actually also three vesicle, sorry, all three products contained vesicle in the range that we're looking for, which is around 150 nanometers. But as we know, being vesicle researchers, only seeing is believing. So we checked with transmission electron microscopy, which was in our case the Philips Techni, counterstain with urinal acetate, whether we can find vesicle structures or if we only find debris. And what we actually found was in all three sources, vesicle-like structures in all the samples that we checked. So with that, we could move on and say, okay, we have vesicles, but now are they having any function? Are they capable of interacting with bacteria, for example, with cells? So we started to assess potential effects of especially royal jelly extracellular vesicles on biofilms and also on cells. For that, we chose a wound-associated pathogen, which is Staphylococcus aureus, which is often found in chronic wounds, which is biofilm forming, which is also an implant-associated pathogen and capable of colonizing metals. And recently, in the last couple of years, methicillin-resistant Staphylococcus aureus, or MRSA, has become endemic. It has become very difficult to control. So looking at the inhibitory and bactericidal effects of especially the vesicles that we isolated, we chose for first assessment a non-biofilm forming Staphylococcus aureus. We bought it from ATCC, which is the strain 25923. And we started to develop a bacterial susceptibility test, which was confirmed with bercesserin. So for that, we encountered our first issues, because usually when we test bacterial susceptibility to a compound, we use concentrations. Sorry, we use concentration. In our case, we could not find a concentration because there's no molarity of exosomes or extracellular vesicles. There's no protein range that you can take. So what we decided was we use ratios of particles that we found to live bacteria. So we started with low ratios, like one live bacteria to one extracellular vesicle and starting increasing that. And what we found was, as you can see here, the metabolic activity is in pink and no metabolic activity is in purple. We found that especially royal jelly is highly antibacterial. So we found that one vesicle is sufficient to reduce viability of 10 bacteria. In the case of honey and bee pollen, it was more like around 1 to 1. And but for royal jelly, it was a factor 10 higher. We then went into biofilm inhibiting effects, which shows a biofilm forming Staphylococcus aureus, also brought from the ATCC, strain 29213, and found that all three, honey, bee pollen, royal jelly, extracellular vesicles, had a strong biofilm inhibiting effect. So you can say that 100 vesicles for one live bacteria was sufficient to not have anything alive. And then with decreasing the concentrations, the ratios, we found an increase in the biofilm formation. So we can say, okay, for bacteria, yeah, we see an effect of those vesicles. But how is it with the cells? Can we see an effect on cells that are involved in wound healing? So we first speculated whether it's actually possible for human cells to internalize bee-derived extracellular vesicles. So we thought of testing that first. And for that, we used a fluorescent dye called CFSE, with which we stained the vesicles and incubated mesenchymal stem cells with those fluorescent extracellular vesicles, and assessed that with confocal microscopy. And as you can see here, you can see those tiny little green dots, hopefully. Yes, human cells can internalize bee-derived extracellular vesicles. When I first showed this at a lab meeting, everybody looked at me like, yeah, but I mean, that's not inside the cell. How can you prove it's inside the cell? It's like, yeah, but I mean, it's around the nucleus, it has to be, right? And I was like, So we actually designed another experiment using flow cytometry. Again, we dyed the extracellular vesicles, we incubated mesenchymal stem cells with the fluorescent vesicles, and then removed the cells from the petri dish and assessed fluorescence with flow cytometry. And with that, you have the cell size together with the fluorescence, and you can be sure that the vesicle has been integrated into the cell. And what we found was that all three types of vesicles have been integrated into human cells. Royal jelly a bit more than bee pollen and honey, but overall there are all around like 20% of the cells incorporate those vesicles. So the next question was, yeah, but are those functional? Because a cell integrates something quite easily and then gets rid of it quite easily as well. So we designed a cell migration assay using extracellular vesicles. But I've used mesenchymal stem cells growing in a plate until a cell layer was confluent, inflicted a scratch, and measured after 0, 4, 8, and 24 hours. Basically showing if you have high migration of the cells, it's something that you actually want in wound healing. And if you have low migration, that would be like the control group. It's an effect that is less favorable for wound healing. So what we found was very interesting for us because you can see here at 0 hours, a nice scratch in all the groups. But after 24 hours, control group and bee pollen do look very similar. You can also see that in the quantification. There is no difference between the control group and the bee pollen group. Bee pollen had no effect on the migration of mesenchymal stem cells. What we found was a significant increase using honey-derived extracellular vesicles, as well as raw jelly-derived extracellular vesicles, which led to a full scratch closure. So, having all those results and a couple of other ones that we haven't published yet, the question was, how do we apply raw jelly Vs on a wound? There's always, of course, the option of having systemic administration, which, looking at bees and also having bee allergic reactions, etc., did not seem like the best the best idea for us. So we thought, okay, maybe we can find a way of having a local administration. And that was actually a side project of my PhD student, Orlando Ramirez, was developing a biomaterial that can release extracellular vesicles. So as a first biomaterial, we chose type 1 collagen, which is ideal for a number of reasons. First of all, it's a natural polymer. So it's biocompatible, it's biodegradable, it shows lower immunogenicity, and it also has a very favorable porosity. So you can see here, that is not our publication, found it in the paper. Collagen of different concentrations has a different porosity, which could be ideal or less ideal for the liberation of vesicles. So thinking, for example, if you have a very low porosity, the vesicles might just be liberated in one day. If you have a very high density, the vesicles might not be released at all. So we tried to test that, and for that we chose different concentrations of collagen gels, which was 1 milligram, 2 milligram, 3 milligrams per milliliter. We took the supernatant that was covering the gels and assessed that with nanoparticle tracking analysis. And what we found, and I hope it's kind of clear, was the following. After 1 day, there's not much release of vesicles. So around 5 times 10 to the power of 6 vesicles being released in a group of 1 and 2 milligrams. 3 milligram basically doesn't produce anything, which also doesn't change over the course of a week. So there's almost no vesicle release in the group of 3 milligrams per milliliter, so the high group. However, interestingly, 1 milligram per milliliter has a peak of 3 days and then decreases. And 2 milligrams per milliliter appears to be an ideal concentration because it increases over time. So the high release after 3 days and up to 7 days. We also actually wanted to see how vesicles are being liberated from a collagen gel, to see the structure, if it changes the structure of the collagen gel, if we can actually see the vesicles being liberated in real time. And for that, we chose atomic force microscopy. For atomic force microscopy, you have a cantilever, a laser, and a photodiode taking the laser signal. And with that, it's actually possible to see nano changes in topography. So that was done in collaboration with Dr. Sebastian Arroyo, which is also coincidentally my husband. And he's an expert on atomic force microscopy. And what he found was that those were collagen gels without vesicles. And with vesicles, we actually found a change in the topography. So we could see that it actually changes the way the collagen is formed. So you can see maybe vesicles within the collagen fibers as well. And we can see in real time vesicles being released from the collagen gel. So we also wanted to know whether the vesicles released have the same functionality as the vesicles that we add to a petri dish. For that, we used, again, the in vitro wound healing assay. And in this case, human dermal fibroblast. We preconditioned the cells, infected the scratch, and then again looked at migration. And what we found was that on day 1, 3, and 7, the royal jelly extracellular vesicles released from collagen gels had an increasing effect on migration. So, concluding my talk, royal jelly, honey, and bee pollen contain extracellular vesicles. These vesicles have been shown to be antibacterial and biofilm inhibiting in their function. They are internalized into human cells. We did find differences between royal jelly, honey, and bee pollen vesicles on human mesenchymal stem cells. And we also found that type 1 collagen is a suitable release system for the topical application of those vesicles. So I'd like you, thank you for your attention, and thank you very much for the invitation as well. So I hope you found it interesting, and I hope we have a lot of questions.
Okay, great. That was an awesome talk. I learned a lot. I know that was really interesting. I have a couple questions, but I'll if anybody has a question, feel free to put it in the chat, or if you want to unmute and ask it yourself, feel free. So I have a couple questions. One is that, like, just from a simple perspective, where do you get the bee honey royal jelly? From the same like one source, or do you use different bee farms? I guess.
Actually, in Chile, we're very lucky because we have a lot of beekeeping here, and we have a lot of the genetic genotype of the bees here is very pure. So we have pure Apis mellifera that we can use for our studies, and yeah, we have different providers that are all organic. And for honey, we have different sources. Royal jelly, we try to not change the source so much because we have one very reliable source, but we've tested now, I think, a total of 38 batches of royal jelly.
Oh wow! Okay.
Oh, yeah.
And then you might have mentioned it, but if you look at the concentration, was the concentration higher? For example, per volume in honey or royal jelly or pollen?
I haven't mentioned it, but I think the purity was higher in royal jelly. The amount was higher in honey. So in honey, since there's also a lot of plant particles involved in that, we find more vesicles, we find more particles. Usually the particles are also more dispersed, but the purity is higher in royal jelly. But taken together, actually, we were surprised by the amount of vesicles we can get out of 1 gram of royal jelly, for example. It's really high. It's enough for like a good amount of assays.
Well, that's great, especially for technically speaking, when you're trying to do all those assays, you need a lot of vesicles. So that's always a challenge in a lot of the work. So great job. Okay, so there's a question from Tanina. Her mic's not working. She said, fascinating work. Did you characterize these BEVs to know which molecules are responsible for the observed effect?
Yes. Yes, we have actually done that. We have not published it because we're just in the process of writing it. So that's why it wasn't in the presentation. But what we found was that one of the main proteins that are involved in royal jelly and honey also is within the vesicles, and that is major royal jelly protein 1. So I couldn't put it on the slide, but I can tell you. Yeah, so major royal jelly protein 1 we found within vesicles. And that has been shown to have a number of effects on proliferation, inflammation, et cetera, et cetera.
Okay, great. So Phil Askanese says, Chen Yang Zhang showed microRNAs are involved in colony individual differentiate. Are the EVs involved?
Okay. If the question is whether those EVs have microRNAs, yeah, we haven't studied the microRNAs yet. It's something that we're doing at the moment. We have been out of the lab during the pandemic for around 5 to 6 months, and now we're slowly starting to get everything up to speed again. But yeah, it was planned for last year, March, and now we're starting to do that. But we have a number of microRNAs that might be involved. There's a number of microRNAs that have been found in roller jelly and in honey in the raw product. But yeah, we're analyzing that at the moment, and we hope we can find some. Because the interesting thing is also that honey and roller jelly have a lot of microRNAs that are very similar to human microRNAs. There are some that are observed around among species, so that might also explain effects on human cells.
Okay. So Carolina asks, wondering whether this effect or all EVs, can you have a control, i.e. the supernatant, whether they have an effect?
We actually have done that in our paper. We have looked at the supernatant deprived of EVs. So basically spin down and look at everything and look at the EVs, look at the supernatant. And we found that, for example, the antibacterial effect is lower. So we know that honey, for example, due to the methylglyoxal and due to other components, has an effect, has an antibacterial effect. But without the EVs, this effect is lower.
Yeah, I think it's also fascinating with the bee pollen. Is there a reason why you use bee pollen? I mean, is it known that they have wound healing properties? Because I don't really know about it. Yeah.
I didn't either. It was actually we actually tried to think of a bee product that has the hypopharyngeal gland involved, but cannot have any vesicles. And so we thought it's bee pollen is perfect because it's dry. It's stored at room temperature. There's nothing really with the storage and everything. In a beehive, it's at 32 degrees. So we thought, okay, no, can't have. Yeah. Truth is, it looks like the vesicles in the bee pollen are dried out by the pollen, which is one of the reasons the pollen compacts. But yeah, bee pollen also has been shown to have very nice effects on burn wounds. So it's been used in burn wound regeneration.
Yeah, and with that collagenase, I think that is an excellent idea that you put it on the collagenase and putting as a biofilm. Is that sort of like, so just taking it simply, I guess the porosity allow the EVs to migrate towards the wound or towards the surface. Is there some sort of like a reason why they migrate?
Well, we think of collagen and basically all the natural biomaterials as something stiff and rigid, and it doesn't change over time. But the truth is that it continuously forms, reshapes, etc., as long as it's not cross-linked. So I think if you would add some cross-linking agent, the properties might change. But since we don't do that, we think that they just diffuse because it's reorganizing all the time. It diffuses out of the gel.
Yeah, that was,
think about, for example, a wound environment. There might also be some proteases, some exudate, etc., that might have an effect on the gel. So we think that the effect that we see now might be even greater in a real wound environment, which was a test.
It's a brilliant idea. I think that's cool. Phil, please go ahead.
Yeah, my question was that I think the Shenyu Zhang laboratory in Nanjing showed that the microRNAs in the pollen were involved in the differentiation of individuals in the bee colony as to whether they be workers or queens or things like that. And so it's hanging that are those microRNAs and exosomes in the pollen, and I would think there may be some in the honey that have to do with the behavior of the colony.
that's a that's a very
So
interesting thought.
it's a huge piece of fruit that's hanging.
Yeah. yeah. That's super interesting.
So
Yeah, I
you got
didn't know
this.
that actually. I know that the bee pollen was activating the hypopharyngeal gland in young nurse bees, but I had no clue that it was actually that has an effect on bee differentiation.
Exactly.
I'm gonna read the paper definitely.
Yeah,
That's absolutely fascinating.
Look for that or email me and I'll try and find it for you.
Yeah, definitely.
Extraordinarily interesting area.
Yeah, definitely. I mean, one of the things that we always think is that this has amazing effects on our wound healing, or it's antibacterial, it's great, but the truth is that it has to have an effect.
That's what we do. That's what we do. That's what we do. But for the bees, in what they do, is what these are having a role in.
Exactly.
Yeah. Thank you.
Okay, so there's another question in the chat box. Okay, so there's another question in the chat box. Tanina asked, some clinics are treating burn patients with honey patches. Is this one of your working perspectives? And did you perform any experiment comparisons using all honey versus just EVs?
We have not used only burn wounds for honey is not on our list for now. We might be going into this direction, but we have a different approach for now. We have not used all honey because the truth is that we couldn't find a concentration that allows us to compare the EVs versus the honey versus changes the media of the cells too much that has a detrimental effect. So we have not tested that yet. And we are curious about the effect of the EVs isolated to identify, okay, this is the effect that we see, and not so much of the honey, because there's a lot of honey research at the moment, especially towards Manuka honey, which we cannot do.
And her other question was, about the clinics or treating burn patients with honey patches. Is this one of your working perspectives?
I said, honey, no. Maybe. For us, we're not thinking about burn patients.
Oh, but I guess meaning like using EVs instead of honey EVs or royal jelly EVs in the patches.
I think honey EVs are not suitable. Honey itself, yes, because it has a lot of other components that are very interesting for burn wounds, and that is like the methyl glycol, the pH of the honey, other components that are, I can't name them. Mm-hmm. And that together has this effect on the burn wound, because burn wounds need debridement, they have a lot of necrosis, etc., etc. I think the EVs alone cannot do. The EVs alone do have an effect on the cell migration, however, not on I do not imagine that having an effect on wound debridement, because a lot of dead cells, necrosis, etc., I think EVs don't have the potential to do that.
Okay, Carolina has another question. It's also interesting whether honey from different plants matter. Is it indicative of plant-derived EV cargo in the honey?
That's a good question. We hypothesized that no. We think that yes. It is known that monoculture versus multiculture or multifloral honey has an effect on the bee health. Yeah, and we think that the bee health has an effect on the EVs. But we think also that the way honey is produced by going through a bee's stomach, going through a lot of enzymatic degradation of the nectar, etc., etc. The plant vesicles might have an effect on the bee, but might not make it into the honey, in our opinion. We actually talked about that a lot. Is it plant particles, is it plant EVs, or is it honeybee EVs? The fact that we see very similar protein profiles between honey, royal jelly, but not in bee pollen, for example, makes us think that it's more the hypopharyngeal gland than the plant EVs. Also because the revenue, the presence of plant EVs in plants is so low, that's basically not detectable. So
there's some sort of like databases on the bee itself, like protein databases and genomic sequence, because I think part of the challenge is getting the right tools. Like in the human study, we have more options because there are more antibodies available, and it's sort of like there's more options that we can do. So what do you think about the future? How you're going to investigate in great detail this bee honey?
We actually found a very reliable provider of custom-made antibodies. We're very lucky. They had published on bee antibodies before, so we found them and we're like, "You have this paper here. You were cited. Can you make those antibodies for us?" And we're like, "Yeah, sure, no problem." So if anybody needs a custom, I think I can't say them, I can name them here, but if anybody needs a custom antibody source that is very reliable, very professional, and just purely amazing, write me an email. I can give you a contact. But yeah, we are investing in a lot of custom-made antibodies. And the problem is that a lot of the bee proteome is still tentative. So people think they found something, but it has never been proven. And that's also an issue that, given the importance of the honeybee for our survival as humans, because it's basically involved in 70% of all agriculture, we don't know enough. We don't know enough of the bees. And especially in the last years, great efforts have been made to change that. But we don't know enough about the bee proteome. We don't know enough about the bee viruses, the viruses that affect bees, for example. And they have great efforts. More groups have to work on that, really. So I can only recommend join the beehive.
Yeah, almost the feeling is like we're taking it for granted of using bees, using honeys, for wound healing, but we don't really know much about it.
Oh, there's, for example, we tried to look for a cell line, a bee cell line, and there's only one. And it's not available on ATCC.
And how about the stability? I mean, in the collagen film, are they stable? I mean, how long can you sort of keep them?
We thought about what is the application, and how long would a chronic wound, for example, be treated with a patch. We talked to doctors and said, ideally 1 day, maybe 3 days. And if it's like not infected, up to 7 days, they would take a wound patch. And that's also the reason why we tested it for 7 days, to see whether we have release for 7 days. And that is what we can prove. And we found that, yeah, up to 7 days, they were stable. The nice thing also about bee extracellular vesicles is that we don't find the great amount of proteases or DNases, RNases in the raw product. So if you use RNase-free, DNase-free, protease-free PBS, for example, there's not much you're introducing. And the stability of the vesicles is extremely high. We've been using some batches for 2 years now. So you can really isolate them, froze them, had them at minus 80, didn't think about them, and then now we're still using them. It's great. And the great thing is when, okay, raw jelly is a bit more sensitive. It has to be cooled all the time, but the honey and bee pollen are at room temperature. They're stored at room temperature. We get them from the providers and we use them for years. And we have the same result. For everybody working with exosomes or extracellular vesicles and cell culture, where basically the EVs degrade by just, looking at them for a second. We're very lucky, really. We isolate and we don't have to worry about batch stability over time. You're not trying to test them, but the EVs themselves are stable over time at different temperatures, because -80 is of course the classical one,
Yeah.
but they might even be stable at higher temperatures.
Yeah. So with the methodology for the isolation, that's sort of high-speed ultracentrifuge. So I guess you're concentrating or interested in the small EVs, but have you observed like larger EVs in it?
No, because we also filtrating before. So we were focusing on the up to 150 nanometers, 200 nanometer vesicles. But it's because we are filtrating before with a 0.2 micrometer filter. So we have not yet looked at it in a different way. Oh, actually not true. We had a lab incident where we once used a 0.45 filter. But the truth is that was for bee pollen, and we had basically the same purity, and couldn't see anything else because all the particles were at the tube. So it was not much difference to using a 0.2 filter or using a 0.45 filter, because in our case it didn't make it to the bottom of the tube.
So I have a quick question. So the biofilms that you use with the collagen, it's human collagen, right?
Yeah. Oh no.
So,
Sorry, it's rat tail collagen.
Rat tail collagen. Okay. Yeah. And then, so do you think the BEVs have like a type of integrin that's binding to the collagen, or do you think it's more of just the physical, like in your atomic force microscopy, that it was just more the physical, proximity, or do you think that the EVs are actually binding to the collagen?
That is a fascinating question. I absolutely have to test this. The first question is, do bees have integrins? Which I don't know. I would assume.
Yeah.
What the question?
Yeah.
That's something I would have to verify.
Yeah.
That's something I have to verify.
Yeah.
But yeah, I mean, if they have integrins that are capable of binding collagen, that could be definitely one of the reasons why they don't all liberate at the same time.
Yeah.
Interesting. Yeah, I'm sorry, I have to write this down. Yeah. Thank you. Who asked this question? Thank you so much.
Vicky, me. That was mine.
Okay, so who has the put in mouth moment? That would be me.
Yeah,
Thank you, Nikki. It's a fascinating question.
I think this is the great thing that we learn from the questions as well, and then for the next set of experiments to do. That's awesome.
The great thing is that since nobody's working with bee extracellular vesicles, and since also there was no conference in the last year, we were really missing input from other people and from different fields. So I'm extremely grateful to get more questions and more things that we have to investigate. I'm so grateful.
So there's one more question in the chat box from Nicole Arnold. Are EVs present in the products that contain honey, like soaps, hair products, lotions, and bread?
I think bee bread is bee pollen. And I think I took a course in making soap. So given the pH that you have the soap in until it ripens, I don't think so. So I think it's a very basic pH. There's a lot of base in the soap as well. So I'm not sure. Lotions, I think it depends. I think any lotion that has a lot of fat content, a lot of lipid content, probably not, because it would bind and destroy that. And the third product was hair products. I think the same thing. So since all the hair products like shampoo, for example, contain SDS, that is known to destroy vesicles. We use it in the laboratory to rupture the vesicles and get the proteins out. So I'm not sure, but the great thing is that raw jelly, honey, bee pollen, bee bread, etc., propolis contain a lot of other things. So it's not just the vesicles. We're not claiming that the vesicles are the sole thing that has the effect, but we think that the vesicles have an additional effect on the efficacy of bee products. We think, for example, in honey, there's many other things that have been shown to have an effect, and in raw jelly as well.
Okay, great. Any other last minute questions? We're going to say, oh, yeah, now we're at 7 o'clock. Yeah. All right. Well, I guess we can close it down. And thank you so much, Christina, for giving her presentation.
Thank you so
I
much
really
for the
enjoyed it.
invitation.
Learned a lot. Yes. And thank everybody for joining, especially too.